talk you or elaborate more on how we can use proteomics in medicine and or in different aspects of biological research. And hi, I'm Abhilash. I'm a postdoc at Vita Lab at UCSF, and I'm currently doing my postdoc on the topic of cancer immunotherapy, where my role is to discover new therapeutic targets that can be targeted using CAR T cells. Okay, so in this presentation, I'll elaborate more on proteomics and how we can use proteomics for different biological questions and to understand the mechanistic insights of various cancers or various other diseases. So in this presentation, I'll cover more about my PhD work, and since this is getting recorded, I did not get permission to show my current work. So if you are more keen or more interested in learning CAR Ts or kind of different aspects of connecting proteomics with the immunotherapy field, feel free to message me or text me in LinkedIn. Okay, so I'll start with colorectal cancer and how we use proteomics to understand colorectal cancer in a better way. So to begin with, colorectal cancer is a cancer of intestine, large intestine, and it's a very deadly disease where the incidence, the mortality is nearly or more than 50% of the incidence. So if you see the chart, you'll see the colorectal cancer, it comes as a like third most common incident cancer, and in case of mortality is the second most. And why we see lot of different, higher percentage of the mortality is because we are unable to screen the disease at a very early onset. And this disease, if you see the percentage of different stages of the disease, the first two, the stage one and stage two, there's a higher 5-year survival rate. That means if we can treat or screen the disease at an early onset and can treat further, we can cure this cancer. But if we are detecting them at a very later stage, then it's very highly unlikely that we treat this kind of cancers. That's why we focused and we found that why so? Why we are able to treat the cancer if they are regionally limited or at the first few stages, and why it is difficult to treat it? It's mainly because the cancer takes time. The cancer slowly grows and it takes time to become sizable or to become seen by the different diagnostic procedures that are available in clinic now. And this happens due to changes in various, gene structures, different mutations that happens. I won't go into details, but if you see the initiation to the metastasis, it takes years to start this cancer. So if we can detect this cancer at this phase, which is generally between that generally happens between 30 to 60 years, and it takes almost 10 years to grow and mature. So at this phase, this disease is not that deadly, and we can treat this disease very well. There were various literature that suggested the stage one and stage two, in terms of clinical practice, they are not very deadly, and that can be removed by surgery only. But the stage three and four, we need various adjuvant therapy along with surgery to treat the cancer, and still we are not sure how to distinguish, there's no clear boundary to distinguish the stage two from stage three. So in this phase, we had different problems. The first one is we have to diagnose this cancer at very early onset if we can help the clinician, even if in like 10%. So our main goal was to identify a marker or a biomarker that can help us diagnose the cancer at a very early stage. The second one is to distinct the colon polyp from the adenoma. So in colon, we always have some kind of tumorous growth. Not necessarily they are cancerous, but they are there. We call them polyp. And when the polyp metastasizes or grows into cancer, we don't have very distinctive targets or distinctive markers that can distinguish this polyp or this tumorous outgrowth from the cancer. So we also had this challenge: can we identify some markers that will distinctively or distinguish these polyps from the cancer? And the third one is the druggable protein candidates. Even the disease at a very late stage, even if we can identify some kind of protein targets, we can find some cure, we can design some drug against it for better management of this disease. So we use proteomics to deal these kind of problems, and we put forth various objectives and various goals to understand the cancer using proteomics. What is the first goal? It's identification of autoantibody signatures from clinical samples for early detection. The second one is to identify some protein biomarker that can tell our cancer clearly from the polyp, which is non-cancerous. And third is to understand some protein that we identified from our previous study to understand in depth role of the protein in cancer metastasis and to identify some potential drug candidate to target this protein. So the first goal where we used the proteomics was a very different kind of proteomics, right? So the proteomics technique actually include a different domain, different range of techniques where we can use microarray to detect the entire proteome. We can use 2D dyes, or we can use mass spectrometer for these kind of studies. So the understanding this technique or kind of elaborating these different techniques is beyond the scope of this talk, but for this technique, for this objective, we used uh, microarray technology to identify some kind of signatures that can be used for early detection of the cancer. So why we use these tools and what are the autoantibodies? So when we have cancers or when we have different kind of biological mechanism going on in our body, it's very hard to see the protein level changes. So we cannot distinguish a cancer from a healthy cell at a very initial part. That's why we are struggling, right? So what best we can do? So as the cancers are genetically different diseases, they alter, they proliferate more, they grow more, they kind of try to survive the natural environment. And at this phase, they do alter the gene or the proteins. We call them mutation. And when our genes or proteins have mutations, our immune system try to capture those proteins, try to detect those proteins by synthesizing some antibodies. And since these antibodies are getting synthesized by against the cells, even though it is cancer, we call them autoantibodies. So that's the rationale. When there's a tumor secretes some kind of tumor antigens, or tumor express some kind of tumor antigens, and our autoantibody or our immune system try to act against those tumor antigens and synthesize or secrete some kind of antibodies. So this happens at a very early stage when the normal polyp or normal outgrowth starts or starts becoming or becoming cancers. So during this tumorigenesis or cancerogenesis, we can see these autoantibodies, and if we can detect those autoantibodies and kind of segregate them from or compare them with the common or healthy cells and make some kind of panel, we can classify this disease from healthy, right? So we use that technique of detecting autoantibodies using some microarray chip to detect or to predict at least the autoantibody response and detect the disease at a very early onset. What we did, so generally these antibodies or autoantibodies are present on serum. There is a kind of body fluid. So we extracted or we collect serum from the colon cancer patient. And also we try to find some healthy controls so that we compare these control serum samples with the colon cancer serum samples. And then we use some kind of chip where there are proteins embedded on it. So these chip or these microarray chip contains at least or around 20,000 proteins, 20,000 recombinant human proteins in duplicates. What we do, we take the proteins and then try to overlay the serum so that even if there is some autoantibody generated against some protein, they will bind to the protein and then will detect those autoantibodies using some secondary antibody. So this is the standard setup, and we use this, and then we compared them with the healthy controls. And when we had this data, these are the healthy controls, we tried to perform some kind of quality controls and took some basic proteins like IgG or some other standard proteins and tried to check the expression level. And we saw there's not much differences in terms of expression of the IgG. So in terms of heterogeneity, while performing experiments, it was negligible. So whatever data we'll get, it will be mainly from the disease and not from the technical problems. So when we performed the statistical analysis, we found a couple of proteins, or group of proteins at least, where we saw a very higher fold change of autoantibody response as compared to controls. In the sum of the proteins were 407, Kc, and Ab, and there are different kinds of proteins involved in different biological mechanism. And then when we saw this statistical data, we were more curious to see the expression pattern of these protein on actual slides. And when we checked, you will see this HC means the healthy controls, and these are the different groups of or different cohorts of colorectal cancer samples. You can see the expression of the protein, coronin 7, then KCNAB, RAP53. These are almost the early markers that helps the cell or that helps the cancer grow more. So when the cancer arises, it does some kind of changes into its system. It modifies itself that helps in the proliferation of the cancer. And these proteins, when we did the biological, and I'll show you that later. And again, we try to validate this protein using parallel reaction monitoring, which is an MS-based targeted proteomics, where we prepared the peptides and tried to monitor the unique peptides of these proteins in healthy controls and in the colorectal cancer patient sample. And we saw a kind of drastic changes in the expression level of these proteins. And we thought the autoantibody generation is mainly because the protein were expressed, more than twofold in the cancer patients. So then we performed some biological pathway analysis, and we saw that these proteins were involved in the initial part of cancer generations, like degradation of extracellular matrix, some kind of adherence junction, and telomere maintenance. These all happens when the normal cells become cancers and becomes uh, starts converting into cancers. So we had this marker. We had this marker before, but what about their specificity? Can we say that these markers are specific to colorectal cancer? So we tried to download data sets across all potential cancers, whatever cancers have these kind of microarray data, and tried to cross-check the intensity or kind of expression of the of this autoantibodies against other proteins, like these are some kind of pituitary cancer, the glioma, meningioma, lung cancer, and also there's some database called autoantigen atlas, where we try to see the expression pattern of these proteins in all this cancer, and you can see this green spot showing this antibody or this autoantibody is expressed only in colorectal cancer and not in any of this disease. So we made this panel through a high selective or high throughput technique that can be now used for colorectal cancer early detection. Although now we are not kind of now we have stopped working on this study, but we are writing some grant based on this to make a panel using using very low density spot microarray and move forward for clinical trials. So this was one case study that we tried to use proteomics, a very different kind of proteomics using microarray to detect early signatures of colorectal cancers. But the other problems, the problem I told you, we have colorectal polyps which are not cancer. And in some cancer patients, the polyp starts converting into cancer. Can we try to identify some prognostic markers that can differentiate this colon polyp from the colorectal cancer so that we can tell which patient is more prone towards developing cancer? So for that, we used mass spec based proteomics, and we performed quantitative proteomic experiments to identify those kind of markers. So what we did, we extracted, we collected the tumor from the colorectal cancer patients. At the same time, we also collected the peritumoral tissues. These tissues, peritumoral tissues means the tissue surrounding the cancers, which is from a very distant region, and it is not cancerous. And then labeled these cancers with some kind of tag that is used in proteomics called TMT, and then quantified the relative expression of the proteins from peritumoral tissues and primary tumors. And this study was done in a paired-wise manner where we matched the primary tumor with the peritumoral tissue. That means from where we collected primary tumors from the same patient, we got this peritumoral tissue so that we can compare this expression of whichever protein we are getting data properly without any biasness. Then we prepared the samples as per our proteomic experiments where we extracted proteins and then digested using trypsin and then made the peptides, then labeled them with TMT tag, which TMT tags are nothing but the isobaric tag, where these tags have six different, this is a six-plex tag, so that the tag have different kind of isobars where the masses will be same. And then we use those kind of tags to tag the peptides, and later we did this mass spec based analysis. And from this analysis, we were able to see a group of proteins which are highly expressed in the cancer group as compared to the controls. See, our objective was to identify some kind of proteins which are only or kind of very highly present in the cancer, and the expression of those proteins in the controls should be very minimal. So we did this experiment and we saw a group of proteins like GCA or gram calcium, olfactomedin, and this ANPEP. And we saw the proteins were expressed in more than at least twofold, log twofold in the cancers as compared to the healthy controls. And when we saw these proteins, we are quite sure that these proteins are specific because the proteins are mainly related to the intestine metabolic process and whatever function the large intestine does. And later, whatever quantitative data we got from this short-term proteomics, we use targeted proteomics to verify or to validate whether our expression is correct or not. And we saw a similar trend in data for these proteins from the targeted proteomics, where you'll see the expression of olfactomedin in controls are very minimal, but in tumors they are very high. And then we try to see if olfactomedin at all can be used as a specific marker for distinction of the intestinal tube or the intestinal or the colon cancer tumor from the healthy controls. So what we did, we collected the human protein atlas data and tried to see the expression of this olfact. Just a minute. So yeah, we collected the data from HPA and checked the abundance of olfactomerin 4 across all the possible organs in our body. And we saw this is a kind of, it's a very distinct protein where you will see the non-specific expression of the protein in some other organs, but they are more prominent in all the part of intestine, including small and large intestine. So if we collect the tumor which is polyp or non-cancerous, and if we are given a polyp and a cancerous tumor, we can check the olfactomerin 4 level and say which is cancer and which is non-cancer. These kind of proteins will help us distinguish between polyp from the cancer. Similar case for the ANPP or alanine aminopeptidase. From the HPA data, you will see it is mainly located in the alimentary canal, including the colon and the rectum, which is more kind of the intestinal tumor, and it is mainly an intestinal protein. And if we had a similar scenario, we can check the abundance of this protein to distinctively tell which of the sample is cancerous and which one is controlled. So this was the goal, to identify some specific marker where we use the mass spec-based proteomic studies to, to identify the expression, the protein level expression of various, protein level expression of healthy and cancer tissues, and identified some proteins that can tell, or that can at least distinctively um, tell us, or tell clinician to diagnose or prognose the disease in a better way. Now the third case studies, or this is the final case studies where I'll tell how we use proteomics to understand the CAC pathobiology. So in my previous two case studies, we use proteomics for, the first one is for the early detection or early diagnosis of the cancer. The second one is prognosis of the cancer, that can tell that can differentiate the cancer tissues from the healthy cells. And the third case study, or the final case study, where we will use proteomics to understand the CAC pathobiology, to understand more about how the cancer, grows better, which proteins are involved in the better growth, and um, involved in the cancer genesis, and can we target these proteins, and what are the ways that these proteins can be targeted to treat the cancer, or at least to manage the cancer. So in this case, uh, we had some hypotheses before. So due to lack of time, I'm not showing those hypotheses, or I'm not building that hypothesis here in this presentation. But we had seen from our previous study that this pathway, or this biological process, that is de novo lipogenesis, is altered in colorectal cancer. And de novo lipogenesis means generation of lipids or fatty acids for the growth of cancer. We know that our cells are made from proteins and lipids and nucleic acids. These are the basic biomolecules that are present as a building block of our cells. So when the cancer grows rapidly, it needs the some kind of process that will help the cancer cells to make its lipids or fatty acids. So cancer uses this mechanism called de novo lipogenesis, where it uses the mitochondrial citrate using ACLY, this is ATP citrate lyase, that makes acetyl coenzyme A from citrate, and then acetyl coenzyme A condensed with melanin coenzyme A to make palmitic acid. Palmitic acid is nothing but the fatty acids that are required to make the plasmamembrane or make different endomembrane systems. So we saw this process is heavily altered and heavily upregulated in the cancer. But what will happen when we try to treat the cancer using some kind of blocker? So we first try to detect this de novo lipogenesis in the actual patient sample. So whatever studies were done, they are mainly done in cell cultures or in some animal models, but there were no clear rationales of these kind of enzymes on actual patient tissue samples. So similarly, as for my second case study, we extracted or we collected the tissue sample and extracted the proteins and then tried to detect the peptides of these proteins like fatty acid synthase and acetyl, ATP citrate lyase, ACLY. To confirm this de novo lipogenesis is highly upregulated here as well. So we try to see the unique peptide of fatty acid synthase and ACLY, and then data parallel reaction monitoring, which is a kind of targeted proteomics, where we saw different peptides of fatty acid synthase are very high in the CAC sample as compared to its matched control. Like from the same patient, the the cancer sample or the cancerous part showed higher expression as compared to the healthy part. And that was the same case for ACLY as well. That validated that this process is very heavily expressed in the colorectal cancer. And when we looked at the literature, we found that this is not the standard way of making fatty acids in healthy cells. How healthy cells make their fatty acids? We use fatty acid synthase, but also we have the triacyl glycerol or these triglycerides, these pool available for our body. So whenever we eat some fat or extra carbohydrate, they get stored in the form of triglyceride. And this TG is broken using some lipases in our body. That lipases breaks the TG to make the dietary palmitate. And this palmitate is used to build our cells, to build whatever the lipids or fatty acids are required. Also, we need a minimal fatty acid synthase expression for this mechanism as well. But in cancer, in tumor cells, they don't have this TG, right? So TG are present in our body, but not in the cancers. So cancer they have to synthesize fatty acids using fatty acid synthase. There's no other way. So now we know that how important this enzyme for the cancer and how important this enzyme for our healthy cells. So if we can block this FASN, is there a way whether we can kill the cancer? So this was our hypothesis to selectively break or to selectively block fatty acid synthase and see whatever happening to the cancer cells. So for that, we tried to find which compound can be used for blocking the fatty acid synthase, and we had three different options, which are selective inhibitor of fatty acid synthase, like C75, orlistat, and cerulinin. And we selected cerulinin to study this because this has more affinity and this selectively inhibits cancer as compared to the other inhibitors. So we performed some cell assays where we treat cerulinin to see what is happening to the cancer cell lines. So we used one popular cancer cell line, colorectal cancer cell line, HCT116, and tried to treat different concentration of this drug, and so there was a dose dependent cell cytotoxicity. That means whenever we try to increase the drug concentration, there is we saw a gradual increase in the cytotoxicity. Similar thing happened for to the migration assays. So when if you see the controls and the treatment, when we made a wound to the cell line and kind of compared the wound healing capacity of control versus cerulinin, we saw a clear differences between them. So after 48 hours, you will see a clear wound healing process in the controls. Versus the versus this. Later, we did a cell cycle analysis where an apoptosis assay where we use the technique which is already developed properly. During apoptosis, we know there's a selective protein called phosphatidylserine, which is generally present in the inner lipid of our plasma membrane, or mainly cytoplasmic phase of plasma membrane that cleaves to the outer side of plasma membrane. So we selectively label that protein using the antibody annexin V and try to check the apoptosis. And from this assay, we saw if you see the control cell, this quadrant, this should tell which proteins or which cell populations are undergoing the apoptosis process, and we saw a clear rise in apoptosis as a result of cell leveling. That shows if you inhibit this fatty acid synthase, since the cancer cells need the fatty acid synthase to survive, if we inhibit, this will lead to apoptosis or cell death, the programmed cell death. In a similar way, when we did the cell cycle analysis, we saw the healthy cell is properly distributed with Z0, Z1, and D2M. But when we treated, there's a rise in the G0 phase population, sub G1 population, and a clear cell cycle checkpoints after this G0 phase. That means when we treat them, cells are not dividing rapidly and they are undergoing apoptosis. That shows that drug has a potential and this can be used to at least block this fatty acid synthase, and that will help manage the disease. So when we saw apoptosis from the FACS assay, it was important to validate the different markers of apoptosis like BEAD, BAX, caspase-9, or cytokines. So these are the different markers which were again validated using targeted proteomics. And you will see the expression of these apoptosis markers were kind of greater than twofold in the treatment as compared to the controls. That showed there's a clear apoptosis as a result of the treatment. But this was not our goal. Our goal was can we know that we'll able to see apoptosis. But when we are trying to treat the cancer, what what are the possible side effects? So we wanted to study this mechanism using mass spectrometry. We know that we'll treat, at least the cell will die, but we wanted to study the different metabolic changes that will tell if this drug is administered to some clinical patients. We should know what side effects the patient should have, what side effect we should expect. So for that, we did a systemic proteomic studies and tried to see different metabolic processes. So at first, we saw that there is alterations in the lipogenesis because we know we are inhibiting the main enzyme of this process. But along with that, we saw a rapid increase in the oxidation of fatty acids. So we know when we are not making fatty acids, what will happen? We will deplete all fatty acids. So whatever fatty acids were present before were all depleted. So the cell started some kind of, this beta oxidation or oxidative fatty acid. And to counter this process, there's a rapid increase in the pyruvate and glycolytic pathways and PCA cycle and oxidative phosphorylation. So now we know that once we treat the cancer, once we at least administer this kind of drug or small molecule inhibitor, we are bound to have some kind of metabolic changes. And if at all this kind of small molecule passes some clinical trials, we will be ready with, ready with some sort of tools or some kit that or cocktails that will help reduce the side effects of a patient. And then we knew that there's apoptosis, but is there some processes this drug is doing? Is there some kind of key biological pathways is getting altered as a result of this drug administered? And we saw there's a rise in the main or key cancer pathways or processes, including this mTORC, all the pathways in cancers, and along with that, some key oncogenic proteins like this CDC42, the RALA, the RAC1, ROA, these are main cancer, main oncogenic proteins. We saw when we administer this drug, we are seeing a down regulation of these key cancer proteins as compared to the healthy cells or as compared to the controls, right? So that suggested us this drug can be used to inhibit fatty acid synthase, which is responsible at least which is responsible for cancer formation or at least the metastasis. And then when and the and this and understanding this systemic effects will help us tackle the side effects in a more efficient way. Also, when we administered this drug, we saw the key component of this VEGF signaling, which is a kind of which is mainly seen when the cancer is metastasized. The main component of this process were getting down regulated. That showed that this drug can be treated in the higher stages of the patient to at least uh, reduce the main or important key signaling in them. Okay, so this is what we got, and when we did a targeted assays after treating the drug, we saw increase in the proteasome machinery, the ubiquitin pathways, and the ATM kinase. So why I'm telling this? We also wanted to learn the mechanism why there is apoptosis after treating the cancer. We know that fatty acid synthase is important for cancer. So if we inhibit fatty acid synthase, the cell will not synthesize more or not proliferate in a more way, but we didn't knew why there is apoptosis. So to study that, to learn that, we had this objective, we had this hypothesis that this pathway analysis, so there is a proteasome machinery activation as a result of the treatment. So is there a way this is leading towards leading towards the protein digestion within the cell, or kind of in situ protein digestion using this proteasome, ubiquitin proteasome complex? And is there a cell cycle checkpoint? So from this, we saw that when we inhibited this fatty acid synthase, there was a double standard DNA break. How we knew that? This protein, this ATM kinase, is a sensor of this double stranded DNA break. So we try to see the expression of the ATM kinase. That ATM kinase generally activates the ubiquitin proteasome complex that arrests the cells in G1 phase, and that generally leads to apoptosis. So that's how we knew that how or what is the reason behind the cell death as a result of fatty acid synthase inhibitor treatment. And that led us that sertraline is an important drug. And although we need some clinical trials to go ahead with this drug, but can we use some already available drugs in medicines which are used for some other kind of diseases, but not for cancer? So we did this drug repurposing and to find some better therapeutic target which can be taken forward right away for the clinical trials. And we found there are some drugs that act in a similar way as sertraline, including this entrectinib, imatinib, and these different drugs that had the similar binding affinity or even better binding affinity, and that binds to the same active site or binds to the same site where the sertraline binds. So now from this study, we know that there's a We know how to manage this disease using different proteomic tools in terms of early detection of the cancer, prognosis of the cancer, and better management of the disease using some repurposed, you know, inhibitors. So this is my conclusion, and the acknowledgement where, I mean, most part of the studies were done in IIT Bombay, where I did my PhD under Professor Sanjeev Srivastava, and I'm grateful to him and all collaborator and my lab members. Thank you.
Thank you, Abhilash. So, so anybody, if they have any questions, they can ask. And but first, there is one question in the chat box. So, what is PPIN analysis? If you can explain that.
PPIN is the protein-protein interaction network, where if we find some protein which is altered in the biology, which is altered in the cancer or in the disease, we try to see what are the systemic level, what are the systemic information we can see, right? So that's why we do protein-protein interaction network to see more influence proteins from of that particular target, and this helps us knowing the disease in a better way.
So there is the
Okay.
next question is from Shuvishna Datto. Have you performed any knockdown experiments to validate the lipogenic pathway? How did you come up with the hypothesis?
Okay, so this one is, see, we didn't do any knockdown, and our objective was not to study the mechanism of this. It is just the byproduct when we did. We were more curious to do it, but our main goal was to see the lipogenic processes, right? And I didn't show the how we build this hypothesis. So when we use this cancer patient data, and when we did this quantitative proteomic experiment to identify the selective markers for prognosis of the disease, we saw this fatty acid synthase, and ACLY is one of key protein which is highly regulated, which is highly upregulated in the cancer. That's why we come up with this hypothesis that these proteins are also important for the cancer survival or cancer, metastasis, or maintenance of the disease or the cancer. And that's how we try to come up with some clinically relevant concept of using some small molecule which can be repurposed right away and move to clinic.
Then a question is from the question is that it is a general question and not regarding the presentation. That is, I want to do non-targeted quantitative proteomics to compare two samples. Can I use QTOF
Yes.
LCMS for that?
Yeah, QTOF can be used, although it is not a very high throughput, and it doesn't have very high resolution. But you can still use QTOF for this kind of experiment. Yeah, but it would be better if you can use some kind of orbiter or timsTOF instrument. Any other questions?
Can you explain HCT 116?
Sorry?
Can you explain HCT 116? You have told.
HCT116 is a cancer cell line that we use for, it's a colon cancer cell line that we use for our study. Okay.
So Abhilash, I personally had a question that so you have shown that uh, the cell death is happening by apoptosis. So cell death happens by a lot of other things also, the necrosis and pyroptosis also. So what according to will be a confirmative assay for saying that a cell death is happening by apoptosis?
So for that, you need to find apoptosis marker. As I showed, there's a set of apoptosis marker can be used. First, you can do flow cytometry to see the expression of phosphatidylserine on outer membrane, which is the phosphatidylserine is mainly present in the inner membrane or towards the cytoplasmic side at normal cell state. But during apoptosis, due to the flippase activity, it comes to the outer side of the membrane. So you can detect this protein to say to show the apoptosis. But apart from that, you can do Western blotting or viral reaction monitoring of several other apoptosis markers like BAD or BCL family protein, BCL2 family proteins or cytochrome C, and there are some other apoptosis marker that you can target to confirm the apoptosis.
And according to your experience, do these apoptosis markers they can validate using targeted proteomics?
Yes. I showed that in my slide, right? You can see you can do that selective. They are little difficult to do, but you can still see the expression on the cancer cells.
So another
You have to be
question is,
very careful handling the cells for this experiment.
so another question is in the chat box that did you come across the role of mucin 1 biomarker role in CRC?
No, that is very distant from my study. Although I saw a set of mucin family protein which is very distinctively present on colon cancer as compared to healthy, but I didn't explore them. Okay, is there any other questions or queries?
I think someone is writing some question. Let me hold for some time. In the meanwhile, I can ask you one general question from this. That one thing is that so you are currently at the UCSF in California. So means so if somebody is trying to go for the postdoc or PhD there, what is the procedure and what have been your experience there, and what is the topic that you are working there right now, and what is your research interest?
Now I'm working on CAR T cell therapy and finding or discovering a high selective target that can be used for CAR T therapy using high throughput proteomics and chemoproteomics strategies, which is a very different and it's a more advanced level of proteomics that we do, where we don't mainly think of the expressional changes, or we try to see the spectrum in a very in-depth way and see a different mass shifts on the spectra and try to see the different PTMs that is present on the spectra. It's more kind of the spectral search and different way of doing proteomics. So so the main goal is to identify a selective therapeutic target. So that's what I'm doing now as a postdoc. And here it's for postdoc, the strategy is very different than PhD. And PhD is you have to follow the organization rules, how whichever organization you're applying. For US, you need GRE or some English test, which is very kind of relative, and some institute may not ask even. At the same time, you need some publications from your whatever course you are in. Maybe as a co-author is fine, but you need to show some output that you did during your masters or or MTech or whichever course you are in. So mainly you need some publication and you have to clear the English level test, which is very different, varies from university to university. And for postdoc, once you are finishing your PhD, the key thing is you have to be you have to be good at communication. You have to be crisp at communication. You have to show your output, research output in terms of publications or patent or whatever way you got, and also come up with a very clear research. I won't say proposal, but it's a CV along with some kind of research interest that you want to join their lab. So whenever you are applying, read their papers properly and try to see what they're doing and how you can contribute them to make their lab better. So these are few things you have to keep in mind before applying.
Thank you. So I think I will take the last question for today. So is bacteriophage therapy would be an effective treatment against colorectal cancer?
I don't see why. I mean, there's no reason why we can't use this. It will be effective, obviously, but then you have to um, do some more research to see its efficacy and make sure it does not have any side effects and some other things. But if you can design some kind of oncolytic virus that selectively finds the cancer cell and lyses it, you can use it. So yeah, this can be used, but it should be oncolytic or cancer lysing virus.
Uh, so thank you. It was a wonderful presentation today. And if you want
Thank
to,
you, Rajeshna.
if you want to share your thoughts before we end it, you can you can go on.
Yeah, this is a great initiative that you guys are doing, and it's good to connect with you. Probably, whenever you think you are ready for the next goal and struggling with some kind of write-up or any other thing, you just feel free to connect with us. And whenever we have some opportunity, also will be I'm very open to share those opportunities. So it's better to build some network like this and help each other grow.
Thank you. So we can end our session here.
Sure. Yeah.